Hi everyone, my name is Jessica Aldstadt, and today I'm going to be presenting on effector triggered immunity, or ETI, in plants. This topic is important because it helps us understand how plants protect themselves from harmful pathogens and how that knowledge can support agriculture and biotechnology. So to start, we're going to give a little intro to plant immunity. Plants have an immune system, but it's very different from what we see in animals. They don't have antibodies, but they do have two main layers of defense. The first is pattern triggered immunity, PTI, which is a general response triggered by common features found in many pathogens. If a pathogen gets past that, the second layer kicks in, and that layer is called effector triggered immunity, ETI. This is a much stronger, more targeted response where the plant recognizes specific proteins or effectors that the pathogen releases. And this is what we're going to be focusing on today, the effector triggered immunity. Mechanisms of ETI. Well, ETI relies on special proteins inside the plant cell called NLRs. This stands for nucleotide binding leucine rich repeat receptors. These receptors can detect pathogen effectors that have entered the plant cell. Once the effector is recognized, the plant responds by triggering strong defense mechanisms like the hypersensitive response, which is a form of localized cell death, and the other one, systematic acquired resistance, which helps protect the rest of the plant. It's kind of like the plant is sounding an internal alarm system and sealing off the infected area. A recent 2024 study looked at ETI in tomato plants when they were infected with Pseudomonas syringae. They found that the effectiveness of ETI varies across different tomato genotypes, specifically between cultivated and wild species. This is important because it shows that ETI is flexible but yet adaptable, and these differences can help in breeding stronger, more disease-resistant crops. It also helps us understand what specific R genes are doing in each variety. So carrying on from that same tomato plant study, this figure shows how different effectors from Pseudomonas syringae affect two wild tomato species. Each plant was sprayed with a strain of Pseudomonas syringae that carried either an empty vector, EV, or one of 6 different effector alleles. You can find those alleles on the top above the pictures. The empty vector is the negative control, and HOPAB1N is the positive ETI control. In the first column, we can see strong immune responses, meaning stunted or dead plants, showing those effectors trigger ETI. However, in the second column, the ETI responses are a little different. There's a strong reaction to HOPAB1N and HOPBJ1B again, but HOPAR1H and HOPAA1Q don't trigger ETI, meaning this species does not recognize those effectors. This shows that even closely related wild tomato species have differences in how their immune system responds to syringae. It highlights the value of screening multiple accessions to understand the diversity of ETI in natural populations, which this could be useful for breeding disease-resistant tomatoes. Another study from 2025 looked at how ETI signals move through the plant. There are 2 important signaling modules. One promotes immune memory and growth, and the other leads to programmed cell death. These are called the EDS1 PAD4 ADR1 and EDS1 SAG101 NRG1 modules. The interesting thing is that while ETI helps plants fight off disease, it can also slow down growth. So the plant has to find a balance between protecting itself and continuing to grow. So here's an image for us to go over. So this image shows how a plant gets its immune system primed after an infection. It's kind of like preparing for the next attack. When a pathogen triggers ETI, the plant starts to get ready for future threats. That preparation is called priming. The key protein here is EDS1, but it needs help from either PAD4 or SAG101 to actually work. So take a look at the left. We see a plant missing SAG101, but it still has PAD4. PAD4 teams up with EDS1, and together they create a fully primed immune system. This is the strongest protection right here. Now let's look at the right side. On the right, the plant is missing PAD4, but it still has SAG101. This combo gives partial priming. The plant has some defense, but it's not as strong. Now, let's look at the very bottom. On the bottom, the plant is missing both PAD4 and SAG101. Even though EDS1 is there, it cannot work alone. So the plant ends up with no priming and stays vulnerable. This shows that PAD4 is more powerful than SAG101 when it comes to building plant immune memory, but having either one of those is better than having none. So learning how this works helps scientists figure out ways to make crops more resistant to disease. Moving on, we have a 2023 research paper that is suggesting that ETI works like a quarantine. So when a pathogen is detected, the plant shuts down the local area by killing off the infected cells. This is called the hypersensitive response. Instead of sending a whole body immune response like humans, the plant keeps the infection contained to protect its healthy parts. It's a smart system that prevents the spread of disease while minimizing energy use. So, moving on, we're going to talk about pathogen strategies to suppress ETI. Of course, pathogens fight back. Another 2023 study showed that some bacteria have evolved effectors that directly interfere with ETI. These effectors can block the NLR proteins from detecting them or mess with the downstream immune signals. This tells us that plants and pathogens are kind of in an evolutionary race. I mean this by, as plants improve their defenses, pathogens are getting better at avoiding them. This is why it's so important to keep researching and updating our strategies in agriculture. A different angle on ETI was presented by Sue and Gassman in 2023. They looked at the role of reactive oxygen species, or ROS, during ETI. These are chemical molecules produced in chloroplasts that help signal danger and destroy pathogens. This tells us that the immune response isn't just happening in one part of the plant cell, but it's a coordinated event that involves energy centers like chloroplasts. Managing ROS is key because too much can damage the plant, but just enough helps stop infection. Understanding ETI is extremely valuable in agriculture. For example, we can use breeding techniques or CRISPR gene editing to introduce strong R genes into crops, giving them better resistance. This could reduce how often we need to use pesticides, which is so good for the environment and our health. ETI research also supports biotechnology efforts like designing custom plant immune systems for different environments and threats. So overall, really good to understand ETI. Looking ahead, there are several exciting areas for the future of ETI research. First, we need to understand how ETI works under climate stress, like drought or heat, because these conditions affect plant immunity. Second, scientists are trying to create synthetic or universal R proteins that work across many species. Another one, there's a potential for using ETI signaling pathways in biosensors, which could detect infections early before symptoms show. To wrap this up, effector-triggered immunity is a powerful plant defense system that detects specific pathogen proteins and launches strong immune responses. Recent research has shown how complex and dynamic ETI is, involving signaling modules, ROS, and interactions with the environment. By applying this knowledge to agriculture and biotechnology, we can improve crop resilience, reduce pesticide use, and prepare for future challenges in global food security. And lastly, these are the peer-reviewed resources or sources I used for this presentation. All of them were published between 2023 and 2025 and offer recent insights on how ETI works at the molecular and practical levels. This is in APEA format. Thank you so much.